From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on July 17th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 101st episode of Matters Microbial. On to the next milestone. Many thanks to every listener, viewer, guest, and supporter. Truly. First, many thanks to podcast listener Blair McLaughlin in New Zealand, who was supportive and thoughtful. Meant a lot to me. Next, a wonderful instructor and maker in Louisiana, Megan Moore, kindly sent me this beautiful piece of work saying what I do more eloquently than I can. I explore tiny worlds indeed. A few weeks ago, I was invited to give a career talk to high school students of the summer science program investigating bacterial genomics, as you can see here. I'll put a link to this fantastic program in the show notes. It must be so rewarding to work with these wonderful students. My undergraduate summer research students work on many projects as they learn techniques and explore concepts. And one topic that interests both of my students, and me, is finding bacteria with antimicrobial abilities, as the Tiny Earth program previously discussed on this podcast does. One student made dilutions of soil samples, and as you can see here, there were several candidates that appeared to be making antibiotics. Interesting to note that isolates from myself and each student seem to have inhibitory activities against Bacillus mycoides, seen here. Some are more potent than others. Here is a wonderful example of how the isolates inhibited Micrococcus luteus. Look at those zones of inhibition. Thank you, Tiny Earth program, for giving me the training and the tools. One of my isolates smelled like potting soil due to the production of geosmin. Remember, folks, potting soil does not smell like potting soil. What it actually smells like is geosmin, which is in turn from microbes. Anyway, that isolate looked like an actinobacterium on a petri dish, as you can see here. Many actinobacteria produce antibiotics, and I'm sure this is another one. We have fun investigations to continue. I so enjoy working with students and turning them into microbial enthusiasts. When it comes to metabolism, many students are stunned at the complexity of chemical reactions and their interrelationships, as seen here. It's daunting. So I remind them that much of life is about moving electrons around. As the late Dr. Albert Szent-Györgyi put it, life is nothing but electrons looking for a place to rest. And this is true enough. We extract high-energy electrons from our food using carriers like NADH. That high-energy electron is donated to oxygen, creating water. So I often tell students in my classes to hold their breaths and raise their hands when their chest hurts. I tell them it's too many spare electrons. But microbes are not oxycentric as we are. There are so many clever approaches to extracting energy in the microbial world. Sometimes bacterial metabolism can actually generate electricity, as seen in this video. I'll put links in the show notes describing these wonderful microbial fuel cells, which you can make for yourself. Which brings us to the subject of today's podcast, cable bacteria, which are truly amazing. This will change your microbial mind. Thus, I'm very happy to introduce Dr. Nicole Geerlings of the University of Vienna in Austria to discuss some work she did during her PhD thesis exploring these amazing microbial entities. Welcome to the Quality Quorum, Nicole.
Hello. Nice to be here. Welcome. Thank you, Mark.
And this is you're very welcome. This is part, I guess, of the international edition, isn't it?
Yeah, I'm in Vienna at the moment. You are in the United States. So.
Many thousands of miles.
Yeah.
So I think that the first thing that you had mentioned to me is that you want to make clear that this work that you're going to talk about today was based on your PhD work where?
I did my PhD at Utrecht University in the Netherlands, and then I collaborated a lot with people from the University of Antwerp. So that's where I did my PhD work.
So that would be Jack Mittelberg, Philip Meisman, and
Yes.
Lubos Polarecki, right?
Yeah. They were my PhD supervisors.
And the most important thing to say at the beginning is you had a very unusual PhD arrangement. Would you care to share that with listeners and viewers?
Yeah. I was after my master's, I got a teaching degree. So I was teaching chemistry for a few years at a high school, so high school chemistry. And then the Netherlands had a program, a graduate program for teachers. So as a teacher, you could apply for the program and then get a grant. And then half of the time that you would usually teach would be allocated to do your PhD research. So I did this for a little bit more than 5 years. So I would teach for 2 days in the week and then work on my PhD for 3 days in the week. And that was a really, really nice combination. I can recommend it to anyone, but the program has been discontinued, unfortunately.
I understand those things happen, but at the same time, I wanted you to talk about it as you just did, because there are many paths to becoming a microbial scientist. And I think that what your path is especially interesting to people. And I want to thank you for sharing it.
Thanks. Yeah, no, it's also interesting because I started out as an earth scientist, so I don't have a background in microbiology. So it's been a strange but fun path.
I like to tell people, and it's really true, that science often progresses with different viewpoints. If you look at the history of molecular biology, in fact, many of those huge names at the beginning were quantum physicists that had left the field. So
it's true.
I think we learn from different points of view is my point. So thanks for sharing yours.
Yeah, no, you're welcome.
So here we go, talking about electrons moving around.
Yes.
And about cable bacteria. So I think that it's very interesting for folks to remember that metabolism isn't about, and you were making a joke with me earlier about glucose-centric thinking, and I agree. No. It's like saying that photosynthesis is about making glucose, and it is not. It's about making everything.
Yes.
Every carbon skeleton out of CO2 is what it actually is.
Yeah.
But the way that we teach things isn't as accurate as it could be. So really, when we talk about metabolism, would you say it's all about moving electrons around?
Yes, I would. Yeah. Yeah.
And so that introduces a couple terms, and some of the folks that listen or watch may not have had chemistry in the recent past or not at all. So we use the term redox a little bit and about half reactions. Would you care to talk about those concepts?
Yeah, sure. When we talk about redox reactions, we actually talk about electron transfer. So you always have an electron donor, which donates the electrons, to an electron acceptor, which accepts the electrons. So in the case of cable bacteria, the electron donor is hydrogen sulfide or sulfide, and this gives the electrons in the end to oxygen. I mean, it's a whole cascade, but yeah. And then this electron transfer generates energy, which can then be used to get a protomotive force or ATP in the end. And that's what the cells use to grow. Huge. Yeah.
I think it's interesting. I use the term quite often, oxycentrism, and the reason it's taught this way, and I've butted my thick skull against this idea for many, many years, is because when you look at, for example, NADH as the electron carrier and oxygen as the terminal electron acceptor, there's a huge difference in energy that could be tapped, and we do, and that's where we place. In our eukaryocentric view, that's where we place a lot of our attention. Microbes, of course, are much more clever and diverse about it, and can really deal with very modest differences in redox potential. But
Yes.
if they can use oxygen, that would be great. And that brings us to cable bacteria.
Yes. Well, cable bacteria are microbes, but they are multicellular. So they are usually they can be very long, up to a few centimeters, and it's just a lot of cells stacked on top of each other into a filament. And all these cells have to work together to get their energy. So some cells are doing the oxidation of hydrogen sulfide, which generates electrons, and then other cells are reducing the oxygen, which accepts the electrons. So they have this spatial division of labor between the oxidative part of the reaction and the reductive part of the reaction, which is very special if you think about it. So I think if you look at slide 4, we can get into how they function.
Please.
So usually, if you have a sediment environment, so they live mostly in sediments. If you have a sediment environment, they are vertically situated in the sediment, not completely vertically, but more or less vertically. And then in the sediment, you have these different zones. So you have the top layer is oxidized, so there is oxygen is present. And then if you go further down, the oxygen is used up by microbes, including cable bacteria. And then there's a zone where you have nothing, which is, or not nothing, but there's no sulfide or oxygen. So this is the suboxic zone, but a lot of sulfide is produced. So you have sulfate reduction, and that produces sulfide, but this is immediately used up. So there's some sort of, it's called cryptic sulfur cycling. And then if you go further down, you have sulfide present, which is produced from sulfate reduction, but it's not used up. So if you look at the cable bacteria, most of the cells, generally up to 90%, is in this suboxic zone. And there they oxidize all this sulfide that is being produced by sulfate reduction mainly. Could also be from dissolution of iron sulfides, which also produce sulfides. So they oxidize the hydrogen sulfide, which creates sulfate, but also electrons. So these electrons they have like a shared periplasm, which has a lot of fibers, electric fibers, and then the electrons go all the way up to the oxic zone, where you have a few cells, like 10% of the cells of a filament, that give these electrons to oxygen. And this is how they generate energy.
I think this is so fascinating, and part of this is a microbial perspective. You're familiar with Winogrodsky columns.
Yes.
And that's basically a lot of oxygen at the top and a lot of hydrogen sulfide toward the bottom. And you get different bands of different pigmented bacteria because you have this countercurrent issue of oxygen concentration and hydrogen sulfide. And at each slice, there are organisms that predominate there. But we're talking about much more diversity in terms of the environmental conditions, and not different organisms. We're talking about the same organism that's adapted to those changing conditions. I did have a question. You talked about these electrically conductive fibers in the shared periplasm?
Yes, shared periplasm.
Are those the same as nanowires that you might see in Geobacter or Shigella?
No. I think the nanowires are like Pili, and I think and they're very they conduct a lot of electrons. They're very conductive. But what we know about the fibers, we don't know exactly what the structure is or what it is. But it's like a protein structure probably, and it contains some sulfur ligated nickel cofactor. Wow. So it has to do with nickel. I'm not an expert in this electron transport. I'm more an expert in the metabolism, but the structure is different from the pili you would see in Shigella or Geobacter.
Yeah, fascinating stuff.
Yeah.
I only say that because issues, and this really worries me. People make assumptions about what they're looking at, say in one of these microbial fuel cells, right?
Mm-hmm.
And in fact, you don't know that it's nanowires there. I didn't say there were cable bacteria, mind you. But what I am saying is there are different ways to deal with the transfer of electrons.
Yeah.
And that's what we're talking about today.
Yeah, and I think in these microbial fuel cells, it can be a combination of these nanowires. It can be like humic substances that keep transporting these electrons, or it can be conductive minerals, or a combination of these things. I think this is also this was the hypothesis when they first came up with these this separation of the redox half reactions. So when they came up with the separation of the redox half reactions, because this gives you a very specific fingerprint. You can also see this in slide 4. So this oxidation of hydrogen sulfide, besides generating 8 electrons, it also generates a lot of protons. So in the suboxic zone, you would see a pH that's pretty low. And in the oxic zone, where oxygen takes up the electrons, it also takes up protons. So you would get an increase in the pH. So you have a very distinctive pH peak in this oxic zone. And then the pH drops if you go down into the sediment in the suboxic zone. And this is sort of the biogeochemical fingerprint of cable bacteria. And when the researchers from a group in Aarhus first discovered this, they thought like, okay, it must be redox half reactions separated in space because nothing else could give us this pH profile, which is kind of brilliant if you think about it. And the first working hypothesis was that it's long distance electron transport, but then by all these pilis or other microbial wires, humic substances, and then maybe electron conductive minerals or a combination of these. So they were very surprised when they found the cable bacteria.
It's funny, we're reductionist by our nature, although that's starting to change in science. And I'm always, I'm not trying to quote Jurassic Park all the time, but life finds a way. And this business of living in a place where oxygen is not at the center of the environment, and yet sticking a wire up to use that oxygen is very much, and I found out from you that I did not come up with this metaphor, like this hugely long snorkel to breathe through. If you wanted to live in the mud, but still wanted to take advantage of oxygen, you would have this enormously long snorkel. And from a microbial point of view, a couple of centimeters is, I need to do the math to figure out what the equivalent would be if I were that bacterium, but it's a huge distance and quite surprising, as you mentioned.
Yeah, I think, Philip Meisman, he has a group in Antwerp working on cable bacteria, came up with this metaphor of a snorkel. And I think he also calculated this. So this would be I would breathe in here and then my microbial partner would breathe out 20 kilometers further. So it's a huge distance for microbes. And this is also, if you look at slide number 5, this also extended the scale of electron transport by a lot, by orders of magnitude, like 3 to 4 or 5 orders of magnitude, because usually if this electron transport happens in a normal cell where NADH gives its electron to oxygen, for instance, this is like a nanometer scale. This is a very small distance. And then you already talked about these pili or these vesicles, like you have this Geobacter, the Shigella, or you have some direct interspecies electron transport. I think this can also be done by by wires or pilis or nanowires. This was on a micrometer scale. and cable bacteria just do it on a centimeter scale. So they are really the world champions of electron transport for bacteria. Yeah.
Absolutely.
Yeah.
So what kind of experiments did folks do to show that there was this long distance electron transport?
I think they had this whole hypothesis that this long distance electron transport was a combination of these conductive minerals, the pili, and then maybe some humic substances. But then they started looking into the sediment, and then they saw these cable bacteria, and you can actually see them in slide 2. So you can see a part of the sediment that is taken apart. So you see on top, you see the oxic zone, and then you see a lot of these fibers. So they saw these tufts of fibers, and they were like, okay, these are bacteria. So maybe they're doing the electron transport. So they had this hypothesis like cable bacteria are doing the electron transport, or it's the original hypothesis that it's conductive minerals and all that stuff. And they did some really cool experiments to show that it was the cable bacteria. So they looked at what is the oxic zone, and then they cut it below the oxic zone, like 1 or 2 millimeters. And then they could see that the pH peak in the oxic zone was becoming less prominent. So the pH was dropping in the oxic zone, and the sulfite was coming up from the suboxic zone, from the sulfidic zone. So the suboxic zone became smaller, which means that the activity stopped. So whatever activity was going on was stopped due to the cutting. They put some filters in the sediment with different pore sizes, and there they could see, okay, it must have been something bacteria-sized, because if the pore size was bigger than 2 micrometers, they could still see it going on. And the coolest thing they did is actually they replaced the sediment from 3 to 8 millimeters with like tiny glass beads that were not electrically conductive. And then they still saw this geochemical fingerprint of the cable bacteria. So the activity was still there, but there was nothing that could conduct it between 3 to 8 millimeters into the sediment. And when they looked into this glass bead layer, they found these cable bacteria. So I really like how they went with it. Like they had a hypothesis, like maybe it's cable bacteria. They had an alternative hypothesis, like maybe it's these humic substances and the electron conductive minerals. But they really showed, at least to me, that it was the cable bacteria. And later on, when we got more of a hold on how to get the cable bacteria out of the sediment, we could also measure the conductivity in the cable bacteria themselves. And the conductivity is super high for a biological substance. It's comparable to the conductivity in copper. So it's very high, as long as it's not in an oxic environment.
That's just amazing to think about. And what I wanted listeners and viewers to see is there were levels that they did to, well, first off, they were trying to show it was due to the cable bacteria.
Yeah.
And there were some obvious physical things that they could try to do. And then there were more scientifically eloquent or scientifically intricate ways to look at it as well. It's important for listeners and viewers know when you're moving electrons around, sometimes that leads to excess protons in one area or another, and that affects pH, the acidity or alkalinity. And so this is very similar to showing how mitochondria worked by measuring pH.
Oh, really?
Chloroplasts too. Yeah, absolutely. It was a famous experiment that I did, and it was not original with me, but I would have isolated chloroplasts. I'd be measuring the pH. I would shine light, and they would pump in the protons and change the pH while you watch, turn off the light, and it goes back.
That's very cool.
I'm going to put a link to that. It's called the Jagendorf jump, which sounds like a dance.
Yeah, I will put a link to the actual paper. This is not this is not my work. Mm-hmm. This is from the group in Aarhus who also discovered cable bacteria. But this paper is I just really think the experiments being done are very elegant. And it also shows that you still need this experimental work in microbiology. It's not just the omics. So it's
Right. Right. I mean, we our tools always run ahead of our brains. And so, there was an old-fashioned expression, I talked about it last week on a podcast, a paper about by I think it's Jonathan Beckwith, with toothpicks and logic you can solve problems. He meant it in microbial genetics, but the same principle applies. As I often tell students, you can have all the fancy machines in the world, but this is the best machine.
Yeah.
Right? So I'm so pleased to hear that. And we'll put that link up there as well. What a wonderful idea. So I like the way that you described how they were able to look at these fibers, these cable bacteria. How exciting. And then you wanted to talk a bit about metabolism. Now, again, I want to emphasize to listeners and viewers, these are not like Thiomargarita magnifica, these enormous bacteria with a lot of cytoplasm. That's not what we're talking about here. These are lots of more moderately sized bacteria sharing things. Is that correct?
I would still, I think Thiomargarita is an extreme example. Like you can see those with a naked eye. If you look in slide 3, you can see some microscopy images of cable bacteria, and you see on the top right side, you see a small sediment column, and they lifted the oxygen zone, and then you can actually see the fibers.
Wonderful.
And then in the lower right side, you can see just a regular light microscopy image. So you can see that it's depending a bit on the species, but a single cell in this filament is pretty big. I would say it's between 1 and 5 micrometers in diameter. So that is still quite visible. I work with ammonia oxidizers now, and they are definitely a lot smaller. So I think they're comparable to more of like the Vegeatoa or something like that. So I would say they're part of the big sulfur bacteria.
And the other part that we'll get to in a little bit is the structure that you see within these filaments. And I guess I would just call it sharing of duties.
Yes.
Right. But you were going to talk about metabolism a little bit with us.
As we saw in slide 4, we see that the metabolism is already sort of a division of labor, right? So you see that the lower part of the cells, like the lower 90% of the cells in the sediment are performing the sulfide oxidizing redox reaction, and then the top part of the cells are depositing these electrons onto oxygen. So they are performing the reductive part of the half reaction. So you have already this catabolic division of labor. And we were interested in how this division of labor was anabolically. So how does this affect carbon fixation?
Yes.
So these cable bacteria are autotrophs, which means they fix inorganic carbon. And there are several inorganic carbon fixation pathways. And strangely enough, cable bacteria use it's the Wood-Lyungdale pathway. I won't go into details because I'm not that much into the carbon fixation pathway itself. But
I was going to say that's one of the weird ones that we very seldom have time for in class. And so that's really exciting because the cable bacteria have this job, and I want to talk about how prevalent it all is later. But the point is, it's not using what you would normally expect among autotrophs. And that's what's really cool to see.
Well, it's I think you can expect it I think it's a pretty common pathway in like chemolito autotrophs. But what is strange is that this pathway is very sensitive to oxygen. So the proteins don't function in an oxic environment, and cable bacteria use oxygen. So this is weird, and so that was already a bit of a mystery. So we wanted to know, like, how do the cells within a filament grow? How is this anabolic division of labor if you look at the different redox zones? Because you have one really oxidized zone, one with a high pH, you have a reduced zone with a lower pH. So how does this look like? So to investigate this, we did stable isotope probing. So what we did is we took 13C labeled bicarbonate, and we added this to the sediment, and then we let them grow for a bit, and then after a certain amount of time, we took the Cabolbacteria from the sediment, and then we analyzed them with nanosims, so nanoscale secondary ion mass spectrometry. This has come up before in the podcast, but I can give you a quick introduction into nanosims. So if you look at slide 13, you can actually see a nanosims instrument. So it's nanoscale, so it has a very high spatial resolution, up to 50 nanometers, I think, if you're lucky. And then nanoscale, and then secondary ion mass spectrometry. So it's a mass spectrometry method, and what you do is you have your sample, and you blast it with a primary ion beam. So in our case, we use a cesium plus beam, so we have a positive ion beam, and you let it go over, you raster it over your sample. And this primary ion beam creates secondary ions, and then you measure the mass of these secondary ions. So thereby you can distinguish between the 12C and the 13C, or the 14N and the 15N, and then you can go back in your analysis, you can say, okay, this cell has taken up this much 13C labeled carbon, so this one was active during the incubation. So this is what we did. So overall, we would get these really nice nanosims images because the cells are pretty big. So you can see this in slide 6. So you can see some characteristic nanosims images of Cabolbacteria after stable isotope probing for about 24 hours. And then you can see the different colors denote the 13C ratio, so the 13C over the total C. And then you can see that a lot of these cells have taken up the labeled carbon. And so they were active during the incubation. And as you can see, if you look at the cells within a filament, they also taken up more or less a similar amount of bicarbonate, which is interesting that these cells are all equally active. So what we saw, which was very interesting, if we look in slide 7, we could see if we looked at cells that we took from the suboxic zone, there was a lot of variation in activity, but they were very active. And then if we looked at cells from the oxic zone, we saw no activity at all, or almost no activity at all. So these cells in the oxic zone are not growing. They're not assimilating any carbon. So this means that in this division of labor that they have, the only role the cells in the oxic zone have is to just get rid of these electrons as soon as possible. Mm-hmm. So either they have some community service for the filament, so they go up and then they go down again, and then new cells have to reduce oxygen, or they really do have, they really have an altruistic thing, and then like they reduce oxygen for a while and then they die, and then new cells have to come up and reduce oxygen again. So we don't know which one it is. We do see a lot more dead cells also in the oxic zone.
I'm really appreciative of this. Now, first off, for listeners and viewers, I'm going to put a link in the show notes to stable isotope analysis, number 1. Mm-hmm. So this isn't radioactive work at all, friends.
No.
Right. And then also this nanosims technique, and I was familiar with it as a way of showing how, for example, nitrogen can be transferred from one type of cell in a symbiosis to another. And this is beautiful, beautiful work. I'll put links to both of those techniques in the show notes. I have a question, and I may not be putting it as elegantly as I should. Mm-hmm. So within a filament, mm-hmm. and I presume that filament is made of something.
Yes.
Right? So the cells are inside that filament. Are they like one above the other, or are they in packed in kind of a, I don't know quite what to say, kind of almost like a helix?
No, it's one on top of the other. So they're like stacked.
That's amazing.
So you have cells that are stacked, and then they share the outside. So each cell has its own a cell wall, and then they have a shared cell wall also. And in this periplasmic space that is shared, you have all these fibers that conduct
Right.
the electrons upwards.
Thank you for that. And
Yeah.
what I'm hearing you talking about is that up in the oxic region, you find things that are not metabolically active and in fact may no longer be alive. But their wires are still working, for lack of a better term. And that's one thought.
It's hard to see if something is dead or alive, right? Even it sounds like such a simple thing to analyze, but it's very hard. But no, I don't know if these cells are dead, but they're not metabolically active.
Right.
We do know because sometimes you have a filament even in the suboxic zone, and one cell is metabolically inactive. But then the cells above are super active and the cells below are super active. So this conductive structure that's surrounding the whole filament is pretty robust, and it needs to be, right? Because if one cell fails, then all the other cells below that don't have the access to oxygen anymore. But they do.
And that's really remarkable. Again,
Yeah.
This is like almost like not one wire, but a whole bunch of wires.
Yes.
In that tiny. So it's really an interesting analog of talking about physical systems. And yes, I know you look at a fine enough level, biology is physics too. But this is like really interesting. And again, and I want to emphasize this when people hear about this, I know that you don't, and I'm not trying to say that you do, but people are a little bit aware of nanowires, and this is quite different. And I'm really fascinated by what they're doing.
Yeah, no, it is very fascinating. And if you look in slide 3, the middle image, you can actually see these fibers. So you see a few cells. You see a SEM image or a scanning electron microscopy image of a few cells. And then you can see like this pattern of fibers on top of it. And that's like each fiber is electron conducting, but then contains several subfibrils that are also very So it's very interesting. If you look at all these scanning electron images or transmission electron images, it's beautiful. It's very cool to see what this looks like.
So my friends that do cryo-electron tomography will talk, for example, about chemotaxis arrays on the surface of cells, and they do look geometrical in a way. And they should. I mean, there's mathematics in everything, as my wife, who's a mathematician, often tells me. But I mean, this is a wonderful analog for electronics.
Yeah.
May I ask a quick question?
Sure.
So when we say cable bacteria, I'm having the impression this is not one particular taxon, but there is more than one type of bacterium that can form these associations?
Well, they're all Desulfobulbacea, so it's but it's as far as we know now, if I'm still correct, because I haven't worked on cable bacteria for 3 years. There's 2 genuses.
Okay.
But they're related. So I think the working hypothesis is that this is like an evolutionary fluke. So this is an event that happened one time.
Right. So monophyletic is the term that I've used in classes when I teach for this, and it kind of makes sense to me because this isn't just one thing. This is a series of things that have to take place, and so it's hard to see how that could happen multiple times automatically. To me.
Yeah, maybe I'm not that much of an expert in this evolutionary track. But it's, I think they've been looking for different ones, but they haven't really been found. It's always, it's always these disulfopyldacea.
That's fascinating
Yeah.
stuff. I have a note to myself to write some more about that. So this is a really and fascinating system. And you find it pretty much where you have this kind of stratified sediment is what I'm thinking.
Yeah, they prefer sediment. Yeah, that doesn't have too much burrowing, so not too much bioturbation. Not too much bioturbation.
It's a great word.
Because
it's a great word.
This would break the filaments.
Mm-hmm.
Although they have been found, like sometimes you have these mussel beds, and then below that you have like a nice layer of undisturbed sediment. So they're there. I think they have been found attached to the roots of plants, so sometimes they're in the soil too. So they have a pretty extended niche, or they create their own niche, right? Because if you have sediment and you have oxygen and sulfite that is in the same spatial zone, so you don't have this suboxic zone, and you get cable bacteria there, they start creating this suboxic zone. So they thereby outcompete like the regular sulfite oxidizers that need oxygen and sulfite to be present in the same space. So yeah, they're pretty, I think they're called ecosystem engineers. They're like the microbial beavers.
Microbial beavers. I love it. I cannot wait to have one of my students do some artwork of that, since I do a lot of that kind of stuff with students. It's just amazing. Oh, I love this.
Well, honestly, I think if you have sediment and you sieve it and unpack it, and then have some oxic layer on top of it, some oxic water on top of it, there's a high chance you will grow cable bacteria, actually, because this is how we used to grow them.
And we have some environments here in Puget Sound where there's a lot of buried wood waste, and so you have extremely high levels of hydrogen sulfide there, and then at the same time there's the surface, and I'm wondering if I might find them there.
Yeah, maybe.
Because I see big fibers all the time, so I don't know what they are quite yet. But wouldn't that be funny and ironic if they were in fact cable bacteria?
Yeah, it could be. Yeah. No, it could be. I think it has to hit the light correctly for you to see them.
Okay.
But yeah.
So I know that Beggiatoa and Thioploca and Thiothrix, things of that nature, sometimes are modal, and and they move up and down within the sediment to deal with that. And the cable bacteria, I guess, just grow up into it, and that's why they could be sheared, is because of that.
I think these are all evolutionary adaptations to the same problem. So you have this electron donor, and you have an electron acceptor, and they are either separated in space or in time. So I think, if I'm correct, is the Thioploca one that's a really big one, right?
Yes.
Yeah. So what they do is they just store a lot of nitrate. So they have like their own nitrate pool, and then they oxidize sulfate with that nitrate pool, and then whenever there's more nitrate, they will store more of it. So there, the electron acceptor and donor are usually separated in time. And then
I see. Yeah.
Yeah,
makes sense.
and then if you think of the Beggiatoa, they use sulfite and nitrate. This is also often separated in space, can also be the result from bioturbation, for instance. But what they do is they oxidize sulfite, store it as sulfur, go up, and then oxidize this sulfur further with nitrate. I think they also store nitrate, and then they go down again. So there it's also separated in space, but they deal with this separation by moving around in the sediment. And then cable bacteria have a new method. So they just stick in the sediment and they divide the labor between the cells. So it's all different adaptations to the same problem.
And if you think about it, it's all about harvesting your electrons to run metabolism.
Yeah.
And that's just so interesting to me. I'm thinking about these filaments that you see for the cable. I'm just going to call them the cables.
That's
so
okay.
The cables, and This is going to sound like an odd question because I know that they're somewhat fragile, but if you could flip them upside down, what would be the effect? How quickly could they adapt? Could they adapt? That kind of thing.
Well, that's a very good question, and we try to answer this question as well. So we didn't flip it. We didn't flip the sediment because that's very difficult, or flip the cable bacteria. That's practically impossible because they're in the sediment. But we had a kind of a different approach, which you can see in slide 9. So what we did is we cut the sediment below the oxic zone.
Right.
And then we kept the slice on, and then we measured the electric field, which is a measure of their activity. So once we cut it, we saw a drop in the electric field. But then, if we take the slice away and the top that was first in a reduced environment becomes oxidized, and then we measure the electric field again, we see that it recovers very quickly. You can see this in slide. So you can see the experiment in slide 9, but then you can see the results in slide 10. So if we cut the filament, we see a drop in the electric potential after 2 minutes, and this stays like this for up to 30 minutes. This is how long we did the experiment. But then if we remove the slice and the cells that were first in this reduced environment get access to the oxygen, we see an immediate shift in the electric potential again. And this is after 2 minutes, and this remains up until 30 minutes. So this is how long we measured. So we cannot do a faster measurement, so it takes a while to set everything up. So, but within 2 minutes, it's restored. So we think this is maybe too fast for gene expression, but it could be gene expression. But we think they have everything always at the ready to go and reduce oxygen once they encounter oxygen.
That's really elegantly put and very clear cut. Thank you.
Yeah.
You've mentioned a little bit about how the cable bacteria are modifying the environment in which they are existing. This comes back, of course, to the microbial beavers idea. So we know what the advantage is to the cable bacteria of this modification. Are there other advantages it might get? For example, does it prevent competitors from growing in the same environment, etc.?
Well, this is a big field of research. Because of this cable bacteria activity, you get this suboxic zone. So this really outcompetes any single cell sulfide oxidizers that need oxygen and sulfide to be present in the same environment. In natural environments, we see sort of a succession of cable bacteria and, for instance, Bergiatoa, although we don't really know how they compete. And then there are a lot of bacteria that are associated with these with the cables. So you can see them swarming around if you look under a microscope, for instance. But what this interaction is, that I don't think they know this yet. It's like maybe it's that these bacteria are depositing electrons onto the cable. There's some evidence for that, but I'm not entirely sure. Their whole metabolism is pretty advantageous for them, because this decrease in pH in the suboxic zone this decrease in pH dissolves iron sulfides that are easily dissolvable, so not pyrite, but just like more amorphous iron sulfides are dissolved. So you get more sulfide, which the cable bacteria can then use again. So they sort of create their own sulfide as well. So you have sulfate reducing bacteria that create sulfide, and then you have, due to the drop in pH, you get more sulfide because these iron sulfides start to dissolve. And I guess sulfate-reducing bacteria in a freshwater environment also have an advantage if cable bacteria are there. So in a marine environment, there's so much sulfate present that the production of sulfate by cable bacteria wouldn't make that much of a difference. But in a freshwater environment, there's not so much sulfate. So if you have oxidation of sulfite to sulfate by Campylobacteria, I would argue you would get an increase in sulfate-reducing bacteria as well.
Very cool. This has been wonderful stuff, and I think people are going to look at sediment in a different way. This idea that the Campylobacteria are like microbial beavers, they're engineering their environment, I think is really evocative, and I think I can promise you, I'm going to really lean on a student in the fall to write a term paper on this, because it's wonderful, and I can't wait for the cartoon that they make of this.
Yeah, because it's also because they did research on this, because of this pH gradient you have. So if you go back to slide 4, you can see that in this suboxic zone where the pH is low, iron sulfides start to dissolve, and then the iron start to move upwards. So it diffuses upwards, and there it finds oxygen again. So you get iron oxyhydroxides. So often if you have these sediments where iron is present, which is in marine systems often the case, not so much in freshwater systems, but in marine systems you often have this, you would get like a thick layer of iron oxyhydroxides on top of the sediment. And then if you have a system where you have like seasonal hypoxia, after a while, you get a lot of sulfate reduction, and there's no Campylobacteria because there's no oxygen. And then this sulfite starts to creep up into the sediment, but then it hits this oxyhydroxide layer. So the sulfite cannot diffuse further upwards into the water. So there it can delay like the onset of euxemia for like a few weeks. So in that sense, it's also nice that you have this thick iron layer that keeps the sulfite trapped in iron sulfides. Which is, I guess, better for all the life in a body of water.
I think this is wonderful, and it really shows that the microbial world and the world that we see have an interesting connection, and it shouldn't be a surprise. I mean, they were here first, right? And they'll be here last, right?
Yeah, definitely.
And so I just find the whole thing, this idea of modifying their own environment in a way, is just a creative one. And the idea of this is just so much stuff to think about. One of my big interests is this subject, socio-microbiology, about how microbes cooperate to do things. Mm-hmm. And I just, this is yet another bit of information in my data bank for this.
Yeah. What I find very interesting is that because I've been looking at very small scales during my PhD, and also now, like I use mainly single cell methods to look at very small scales, but the consequences of what these microbes are doing are like on a very large scale, like they affect biogeochemical cycles. So Campylobacteria can have a big impact on the sulfur cycling within a sediment. Which also affects the iron cycle and the calcium cycle and maybe manganese. So it's all connected. So what is happening on this very small scale has effect on this really big scale, which is super fascinating.
For me, when I was an undergraduate, a challenging course for me was ecology, because there was so much complexity to it. Mm-hmm. It was so theoretical. And at the same time, we would hike out and look at this environment and that environment. And the more that I look into the microbial world after all these years, the more that I see that it's all about ecology on every level.
Yes.
And to me, that's like ironic. But I'm loving it. Can you say a few words about what you're doing now for our listeners and viewers?
Yes, I now work on complete ammonia oxidizers, which were also pretty recently discovered, about a decade ago. So there's still a lot to discover, I would argue. And I look at division of labor into complete ammonia oxidizers. So to see if you have subpopulations within a population that either cooperate, or if you have a biofilm, you can think of a subpopulation that makes all the EPS. So if this is happening or not, and when this is happening, what are the conditions that create these subpopulations, that kind of stuff. So this heterogeneity within microbes of the same population is very interesting to me.
It's also something that every geneticist I know cringes at, because they want to believe that all members of a given population of microbes that they're studying are the same. And I've always been nervous about that. And it turns out that I'm right to be nervous about it, because there's, we project our own feelings onto the microbial world, and they're in business for themselves, not us. And,
No,
go ahead.
Yeah,
please.
no, I completely agree. I think it's also, if we look at a genome, there's so much we don't know.
Yes.
And then, for instance, if you look at cable bacteria, if you look at the 16S rRNA or the genomic makeup, you would think it's a sulfate reducer, but it's a sulfite oxidizing bacteria, because the pathway is the other way around. So it's, again, you really need to do this combination. You need both. You need the omics, but you need the experiments in the lab. You need to see what is happening under certain conditions that are preferably environmentally relevant.
There's a paper that was just published, and it's Brett Baker, and this is about how when you look at microbes, you have to look at them not in the context of the laboratory, but in the context of their environment. And I'll put a link to that paper in the show notes because it really impressed me. Because you couldn't study human societies by picking individual people out and studying them. If you were an extraterrestrial, you would want to look at groups of people. And even then, depending on the sample size, you'd have problems. So it's the same kind of idea. And please.
Yeah, no, I completely agree. Is this the same paper that asked for a new model organism?
Yep.
So we would, I completely agree with that paper. That's a very nice publication.
No, I was really struck by it because I've been saying it for some time. I mentioned a little bit about centricities, oxycentricity, eukaryocentricity. And sometimes microbiologists think that all microbes do things the way E. coli does. I call that coli centricity. And darn it, all organisms are not E. coli. And as someone told me, and even E. coli isn't like E. coli sometimes, which I loved.
Yeah, and I think if you think about E. coli, this is really an organism that likes a rich environment, but most environments are not rich at all. So this is, it's very important to also look at oligotrophic environments, I would argue.
So as we start to wind down a little bit here toward the end of our time together, what are just a couple of things or a few things that you would like listeners and viewers to remember about cable bacteria?
Well, I think just overall, they're super cool. They're multicellular, I would argue. I mean, we can have a discussion on that, but they're multicellular. They have to work together to get to oxidize the sulfite and to grow. And I think it's very important to know that bacteria can have a big impact on an environment. So their metabolism can have a big impact on the environment. And there's a lot we still don't know. So I think that is the main message, I would say.
No, and I think that that's one of again, we tend to think about bad effects that some microbes can have on us. We're starting to look about the effect we can have on microbes, and this is the same idea.
Yeah.
So rather than look at an organism by itself, the context of its environment is really important as well, and the effects that both have. It's very much like this.
Yes.
And then I guess finally, was there one experience in your life that made you think that turned you into a microbial scientist?
Yes. Well, it's not one, but there's a few instances where okay, I am working in microbiology now. I think the first time is when we got the first results back from the nanosims, because the original idea was to look at cells that were encrusted. So sometimes these cable bacteria cells have like a crust of iron oxides on it, and we wanted to know, are these cells metabolically active or not? And these encrusted cells are also always in the oxic zone. So we wanted to compare cells from the oxic zone with cells from the suboxic zone. And when we got the first results back from the oxic zone, we thought like, oh, the experiment failed because we don't see any uptake of carbon dioxide. And then we saw the uptake in the suboxic zone. So we got really enthusiastic. But then, I mean, we had to do a control experiment to be absolutely sure that there's bicarbonate in the oxic zone. So then it was like, okay, I'm not looking at the environment anymore. I'm looking at the bacteria now. This is microbial ecology, what I'm doing now. It's not earth sciences anymore, or interactions between bacteria and the sediment. It's really, I'm just looking at bacteria now. And then I came to Vienna almost 3 years ago now. This is a microbial ecology department, so in the beginning I was like, oh, I don't know anything. But now I think I'm a microbial ecologist now.
So the reason that I bring this up is that I run into a lot of students who think there's only one path to becoming a microbial scientist. And if nothing else from this podcast, there's a huge amount of diversity in approaches. And it all depends on what really motivates you. I mean, what it is that makes you want to go in the next day or see what the next experiment is. And that's what makes a career in science, I think, so exciting, despite all the worries that all of us have, is that we get to find out things that no one knew before. I didn't say they were super important cosmically, but you were part of uncovering it. And it's a wonderful feeling.
Yeah,
Nicole,
sorry.
Not at all. Please finish your thought.
The discovery part is very excited that you're like, I was doing this with a master student, and then we're like, oh, we're now the only people who know this. And then you go to your supervisor and you tell them. But that's very cool. Yeah.
That's something to put in your pocket and metaphorically and hold on to. Nicole, I want to thank you for your time today. I've always been interested in bacteria since they were first touted. And I'm excited to have had a Nicole-based view of the bacteria. I really want to thank you for your time. I have never been to Austria, so someday I hope to be able to see Vienna and see what that's like. But until then, I wish you and yours the very, very best.
Thank you so much. Thank you for having me. Because I came here in Vienna and I was like, oh, I'm not a microbial ecologist. I don't know that much. I started listening to podcasts, and this was one of them. So I'm a fan. I learned a lot from this podcast. Also, that it takes many paths to become a microbial ecologist. So thank you so much for having me. I learned a lot.
Thanks. Thank you for saying so. Best wishes.
Thank you.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with epic links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Nicole Geerlings is a postdoctoral scientist and can be found in the Center for Microbiology and Environmental Systems Science, Department of Microbiology and Ecosystem Science at the University of Vienna in Austria. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.